Okay, good morning. Welcome to the penultimate lecture in evolutionary robotics. We will get back to talking about Xenobots in a moment, but let's talk final project. Your fourth and final weekly milestones are due by 11:59 p.m., and then you've got a little bit of breathing room until 8 days from now. Next Wednesday by 11:59 p.m., you will submit on Brightspace your written report and your oral presentation describing your final project. We're going to talk about what that written report and oral presentation is going to be in a moment. All good about weekly milestones? Everyone's on track? Preliminary A/B testing? Okay. All right. So you'll notice now on row 37 here, there's a hot link to written report and oral presentation. Right after class, I will put stubs up on Brightspace for you to submit your written report and your oral presentation. And those stubs will also point to this document. So we're going to go through the written report and oral presentation. Both of them are worth 7% of your final grade each. 7% for your written report and 7% for your oral presentation. That is incorrect. That date should be Wednesday, May 6th. Okay. All right, submitting to Brightspace. As I mentioned, I'll create a stub on Brightspace for written report. You're going to be submitting one PDF document. It should be about 4 pages, double-spaced, 12-point font. It's not onerous. You're not writing War and Peace. You're summarizing verbally and pictorially for us what you did in your final project, which is basically A/B testing. So I'm going to very quickly read through this document just so we're all on the same page, literally and figuratively. Okay. At the moment, hopefully you're finishing up preliminary A/B testing, where on the horizontal axis you have evolutionary progress, usually, or evolutionary time, usually measured in generations, and on the vertical axis, fitness. Whatever it is you're trying to get your robot to do, more F is better. Preliminary test, A/B testing might look something like this. Is A better than B? Is B better than A? Is there no real difference between A and B in terms of how easy or how hard A or B make things for your evolutionary algorithm? You cannot know with one run. So between now and next Wednesday night, you're trying to collect, you're basically doing data collection. Hopefully you've finished all your coding and you can just let your device churn away on many, many independent runs of your evolutionary algorithm, so that at the end, you might actually know, it might not be a lot, but there might be a small difference between A or B. Whatever A is or whatever B is, B made things slightly more difficult on your evolutionary algorithm in terms of evolving whatever behavior you wanted your robot to exhibit. That's what you're trying to do between now and Wednesday night. Okay, by running multiple runs of your A and B variant, some of you might also have C and D and E. That's also fine, but just make sure you get enough data to be able to tell the difference, if there is one, between these different variants of your algorithm. Okay. You're then going to generalize some visualizations to visually prove which, if either variant, produced better robots, or whether there is clearly no difference at all. Perfectly fine. What we don't want at the end is, I did A and B, but I don't have enough data to tell which is which. Yeah? Okay. As you're performing your A and B testing, you're going to write all of this up as a report as follows. The report should contain 4 sections, about 1 page per section. It's okay if some sections are less than a page or more than a page. We've got 59 students in this class, so please, not much more than 4 pages. Okay, first section should describe your goals. What project did you tackle during the deliverables? How did you create A and B variants from your overall goal? And what is the question you're trying to answer? Some of you, if you chose a final project from the subreddit, there's really not much to say here. So again, it's okay if this section is a little bit shorter. If you came up with your own idea, you might need a page or at least a page to describe exactly what you're trying to do here. Second section, you're describing implementation details. How did you go about coding up your deliverables and your A and B tests? What was your strategy? How did you break things down in the way that you did? Why did you break them down into those baby steps rather than some other baby steps? We want to hear about your process here. So please describe your process. This is not the place to copy and paste code snippets. That's not very informative to us. Okay. Third section, arguably the most important and the most interesting one, is where you're going to provide your results from the A/B testing. Most importantly, you should demonstrate that evolution did in fact occur. Throughout your final project and the assignments, you might have noticed that at the end, sometimes the best robot with the highest fitness is really not that more fit than some of the initial random robots. So one of the first things we're going to be looking for in your report, is there an appreciable increase in fitness between your initial random robots and your evolved population and your evolved populations? Presumably you're going to spend a fair bit of time talking about your evolved robots. Are they actually evolved? Are they improved at all over what you'd get by just randomly generating synaptic weights or leg lengths or what have you for your robot? First thing we're looking for. So however you visualize your results, make sure it's very easy for us to evaluate that. Okay. Then we're hoping you'll be able to show that one variant did better than another, or that both produced about the same quality of robot. It's perfectly fine if your conclusions are A is no better than B. Fitness curves are probably going to be your best friend here. We would definitely like to see some of those, but we've seen lots of different ways investigators go about visualizing evolutionary progress, so feel free to go back and have a look. I've just reminded you of some of them here: footprint graphs, phylogenetic trees, visualizations of how robust evolved robots are to new environments. If you remember back to the minimal cognition experiments, those investigators often took one of their best evolved robots and dropped it back into situations that that evolved robot and its ancestors had never seen before, and we want to see how well it does. The overall goal of this course is to try and move us closer to autonomous and safe machines. The safety part comes from being able to put an autonomous machine into a new situation and for it to do more or less the right thing. Yeah? Okay. These are all sort of optional, but it'd be nice to see some. Okay. Screenshots of the robot in its virtual environment. If you've got a very different environment from the default flat terrain, you might want to show us some pictures of that. Grades will be deducted if it is unclear whether A was better than B, B was better than A, or A and B are equal. I keep hammering on this. Make sure you get enough data between now and then. Okay, fourth and final section of your written report. Please demonstrate to us that you've thought carefully about your deliverables and your A/B test. What was surprisingly difficult? What was surprisingly easy? What did you learn along the way? Obviously, in the next 8 days, given this time in the academic year, you don't have a lot of time, you don't have a lot of bandwidth. So if you had another year to work on this project, how would you expand your project? You'd probably fix a lot of the wobbly parts of PyroSim and PyBullet and so on. I know I would. What new features would you want PyroSim to have to achieve this? Which aspects of the expanded project do you think would be relatively easy? Which would be difficult? How big a supercomputer are you going to need to carry out this hypothetical larger experiment? Obviously, your final project as a whole is a baby step, right? You're showing us potential for something much bigger. Feel free to tell us your vision in this fourth and final page. Okay, that's the written report. Hopefully not too onerous. Questions, comments? All good? Okay, oral presentation, like your written report, also worth 7% of your final grade. Also due 11:59 p.m. next Wednesday night. There'll also be a stub in Brightspace for you to submit your oral presentation. What are you submitting in that Brightspace stub? You're going to be submitting a link that points to a public YouTube video. Very important that it's public, because if the TA or I can't open it, we got a problem. Okay. Your video is going to be exactly 3 minutes and 25 seconds long. 0% on your oral presentation if it's shorter than 3 minutes and 20 seconds or longer than 3 minutes and 30 seconds. These seem like some arbitrary number. The reason why is when all 59 of you submit your 3.5-minute videos, after midnight on Wednesday, the TA is going to take all 59 videos and stitch them into one long YouTube playlist. We're all going to be back here together next Thursday at 7:30 in the morning. I don't pick the exam period. You can blame the registrar for that one. We're all going to be back here at 7:30 in the morning. I will be up here as usual, and I will press go on the playlist. And that playlist is going to run for, you've guessed it, exactly 3 hours with 2 10-minute breaks built in. We're not machines yet. We're still human. And we'll be done. Okay, so what is your 3-minute, 25-second video going to have in it? Like the report, the oral presentation should contain 4 sections. So the oral presentation is basically a video and oral complement to your written report. Same structure, different presentation style. Okay, 4 sections. Part of your grade for the oral presentation will be your clarity of oral communication. Okay. In the results section of your video, so skipping ahead to section 3, you'll likely want to show a video of an evolved behavior and a video of an evolved behavior and a video of a random behavior. You're going to be showing this in the written report, but it's very hard in a written document, obviously, to visualize a simulation. Always better to show than tell. So one of the things we want to see is, here's what my robot in my environment with my fitness function and my neural network that I built into it, this is what it tends to, here's what a random robot in that situation tends to do. And then the second short video, here's what an evolved robot does. Like I mentioned here, that one of the things the TA and myself would like to see in your oral presentation is that it's obvious there's a difference in these behaviors. If you're evolving for rightward travel, we should see a random robot that more or less bounces on the spot or doesn't move at all. And in the evolved one, we immediately see it moving to the right. I showed you some videos from past years, and without even telling you what the fitness function was, hopefully in most of those videos, you knew exactly what the robot was being evolved to do. That's what we're looking for. Okay. And then you, after showing a random and evolved robot to convince us through videos that evolution actually happened, you'll then show a second pair of videos comparing your best robot from A against your best robot from B. And it might be more difficult in the second pair of videos to communicate to us and your peers that there actually is an evolutionary difference between A and B. It depends on how large, if any, gap there is between your A results and your B results. So far, so good? Okay. Ensure that your video has no sound. The reason why is in this playlist, when your video comes up, you're going to come up to the front of the room and you're going to talk over your video as it plays. So you are the sound for the video. If you don't have any screen capture or video creation software, we're not giving out any Oscars in this class. Video quality isn't really that important. Obviously, you're not going to have a lot of time this week and next week. We're all going to be very busy. So don't put tons of time into making a beautiful video. It's just got to be clear enough for us to see whatever it is you're trying to communicate to us through video. Okay. You can record your screen with your phone. You can download Open Broadcasting System. It's free. It's relatively easy to do video creation. Whatever you want, that's fine. Okay. No points will be deducted for poor video as long as we can see what we need to see. So if the video is so poor that we cannot see whatever it is we're supposed to see, points off for that. Okay. As I mentioned before, make sure that before 11:59 p.m. next Wednesday night, you upload a link to your video, upload your video to YouTube. Be sure to make the link public, and then copy and paste the resulting URL to your Brightspace submission. Okay. More logistics. I've already mentioned this. The TA is then going to stitch all the videos together into a single YouTube playlist. The order of presentations is going to be random, and I'll put up the order on the board early Thursday morning. So when you come to class Thursday morning, you'll see where you are in the order. This is very, very important. The poor TA is going to have to stay up very, very late on Wednesday into Thursday morning. So if you're even a few minutes late, by the time the TA gets to your name in adding your video to the playlist, if the TA cannot find your submission on Brightspace, 0% for the oral presentation. We're just going to skip you in the order. Yeah, there's 59 of us. This train is we're going to run a very tight ship here. If you're not on board, you're not on board. Okay? All right, you heard it here first. All right. Okay. Then, 7:30 in the morning on Thursday, I will bring coffee and donuts for everyone, so you will be guaranteed of your sugar and or caffeine fix, whatever you need, so you'll be ready to go. As I mentioned, at that time, I will start playing the playlist up here at the front for all the student-submitted videos. Okay, I'll make the oral presentation schedule available in the course schedule. I'll drop it right down here. Early Thursday morning, I'll write it up here on the board when everybody comes in, so you'll know exactly when you're presenting. Okay. You may refer to it on your electronic device in class, so we'll roll back our no devices in class rule at that time. Okay. When the student, before you begin speaking, that student is going to stand on this side of the board. When that student begins speaking, you're going to go and stand on the other side of the screen, so you're ready to go. I'm not stopping the playlist video. We got 59 presentations to get through in 3 hours. So again, we're running a very tightly choreographed ship here. Okay. When your video starts, you come to this side and you start narrating. The student that just finished goes and sits down, and around we go. Okay. In order to keep on time, all the videos will play one after the other with no pause in between. However, two 10-minute breaks will be inserted to give everyone a chance to rest. This means when your videos start, you should start speaking immediately. If you run long, I keep meaning to get a big hook, but I don't have a hook yet. If you run long, your video is going to end, and the next one is going to come up, and the next student is going to start speaking and approach you. That will be your cue to sit down. So you need to speak for exactly 3 minutes and 25 seconds. The good news is, because it's so short, you can practice over and over again. Years and years ago, I had to give a talk at MIT for 90 seconds. My wife and I drove from here to Boston, and I practiced that talk in the car the whole way down. My wife could probably give that talk today. She never wants to hear that 90-second spiel again. So practice, practice, practice. Shouldn't take you long to practice, like 20 times in an hour. Get it down pat. There isn't time for ums. I meant, can you please rewind the tape? This is all going to run live. Practice, practice, practice. Okay. All right. Questions. It would be great if we had time for a Q&A session. We don't. So due to time constraints, there won't be any time to field questions. However, please, during the 2 breaks, if you find a project particularly interesting or you have a question, go over and ask your fellow student about it. I've seen the results already. You're all working on really cool, very different projects. A lot of interesting ideas there. You're all putting in a lot of effort. One of the most rewarding things as a presenter is when somebody asks questions afterwards. So if you have one, please do ask your fellow students. Okay. All right. Collegiality. 7:30 in the morning for 3 hours. There's going to be a temptation to tune out during the presentations, as we all have many demands on our attention at this time of year. I know you're all busy. I know there are other classes other than this one, at least vaguely. Okay. However, each of you's put a lot of work into these final projects, and there's a lot of creative, amazing work you're going to see next Thursday. So please practice collegiality, extend your fellow students, especially those that are presenting towards the end of the 3-hour block, the respect they deserve by watching and engaging with their presentations. Okay. This includes staying until the end of everyone's presentations. Again, I know everybody's busy. I've got coffee and donuts. There's going to be cool robots. What else do you need? Okay. There you go. All right. Throw it a lot at you, but I want to just make sure expectations are clear. Any questions about any of that? Yes?
For the A and B testing, how many generations should we be aiming for?
That's a great question. So how many generations on the horizontal axis should you run for? I have absolutely no idea. It depends on how easy or hard your task is for your robot given your evolutionary algorithm. Here's my advice. There's lots of different ways you can do this, and this is part of the fun between now and next Wednesday, is my suggestion would be do a bit of a sanity check. Set up your code to run for just 10 generations or 5 generations, and do one run of A, one run of B, one run of A, one run of B, another run of A, another of B, another of A, another of B, another of A, another of B. If you get this result, lucky you, you're done. That's it. A actually does do better than B. With 10 generations, even on the simplest tasks, I doubt you're going to be so lucky. But if you are, you're done. More likely, you're going to see something like this. You've probably already seen something like this. Yeah? With 10 generations, actually, you're probably not even going to see a difference between the random and quote-unquote evolved. So go back and change just one line in your code, which is how long your evolutionary algorithm runs for, and run it again. Now, maybe, again, if you're lucky, you might start to see A and B separate. But make sure you get enough. You get many of these short runs. If you don't, go back, start again. Make sense? Question?
Yeah, how many tasks should you be doing? Like how many?
Yep.
How many? What do you think is a good number?
I have no idea. In statistics, the magic number is usually 30 of each. If you really want to say something statistically significant, I don't know that you need that many. I can't give you a number because you're all working on different things. The suggestion is don't put all your eggs in one basket. Start short. Run short runs, but a lot of them, to be able to really convince yourself there absolutely is no difference yet between A and B that I can tell. And again, that might be it. You might never see a difference between them, but at least you have that in hand. There is no difference between A and B. Move on to a longer evolutionary run and keep iterating. If there was a hard and fast rule, I'd give it to you, but there isn't. This is the nature of research.
And then how are we going to know our order for the presentation, like the video?
Oh, you just submit your video. We'll stitch it into the playlist at the right order. Everyone's been assigned a random number in the order. You're going to be 17th to present. It'll be up here on the board when you come in Thursday morning at 7:30.
Okay. Oh.
Other questions? No? Okay. All right. Good luck. Back to lecture material. So we're evolving bodies and brains of robots. The whole point of the field of evolutionary robotics, we want to simultaneously optimize the physical structure and the neural controllers of our robots. And we started back in lecture 20 with hopefully now very familiar rigid robots. Joints, rigid assemblies, sensors, motors, neurons, synapses, all the rest. Then we moved into Jello robots, where things got a little bit less familiar. We looked at differentiable robots, where we threw the evolutionary algorithm away altogether, and we used gradient descent, so we could make non-random changes to the physical structure and the neural controller of our robots. We spent a fair bit talking about evolving the physical structure and neural controllers of robots within robots. And then last time we started to talk about a very new class of robots in which we throw all technological components out the window. And now our evolutionary algorithm is trying to design robots not built from metal and plastics and ceramics, but biological materials. The first time we did this back in 2020, this became known as in popular culture as Xenobots. So I started last time by telling you the origin story of Xenobots. I've spent a lot of time thinking about and optimizing the physical structure of machines. We ended last time with this surprising study from my biology colleagues at Tufts, in which a functioning eye grew on an adult frog in the place where functioning eyes tend not to grow. There's lots of lessons you can draw from this particular study. One of them is that animals, and not just plants, seem to be much more amenable to suggestion. You can convince them to take very different forms from what they naturally take in nature. Given this, my biology colleague at Tufts, Michael Levin, reached out to me and said, listen, I read about your evil starfish and the fact that you kind of rearranged the body of your robots. We do the same thing with tadpoles. Why don't we get together and see if we can do something together? We pitched an idea to DARPA, which is the research wing of the US military. They were interested. They funded us. We got together. We had some money. And at the beginning of this research project, this was back in about 2018, we had a weekly Zoom call with our colleagues at Tufts. Us as the roboticists, them as the biologists. The first thing we needed to do is sort of understand what either team could do. What was easy, what was difficult, what was impossible for either group, and we wanted to then find where we could get together and do something together. So you've already seen this robot before. On one of the very first Zoom calls that we had, it was our turn, the roboticist turn, to present, and we showed this and explained basically evolutionary robotics. We explained that we could optimize or improve the physical structure and the controller of this machine. After presenting our work at that time on the Zoom call, the next week on the Zoom call, it was the turn for the biologist to present. And Doug Blackiston, who did the frog eye experiment I just showed you, he showed this slide to us that week and explained that he had built this over the past week because he saw this thing. What is this thing? It's not a Xenobot yet. Obviously, you don't know exactly what this is, but there's a few hints.
I mean, it just looks like a replica of the simulation.
It's a replica, right? So, first of all, I had no idea that this was possible. So Doug explained that he was very competent at ectopic surgery with frogs. Ectopic surgery means basically Frankenstein, moving different parts around. So inspired by our video, Doug extracted some frog skin cells from early frog embryo and carefully put these skin cells together to create this 3D structure, which it's a little difficult to see here, but it's basically floating in room temperature fresh water in a petri dish, and we're looking down through the microscope with Doug. So this little replica is about 1 millimeter across. It doesn't move, it just floats. Okay, once us roboticists understood what we were looking at, what do you think our next question for the biologists was? Does it move? The answer is no. So our next question was, can you make it move? Exactly. That's how the Xenobots got started. So it took Doug about a week to make this. To get this thing to move took another 6 months, but that became the project. We had actually pitched a very different idea to DARPA, the research wing of the US military. I emailed them the moment after this Zoom call finished, and I said, listen, we've got a different idea. What do you think about the following? What a surprise. DARPA said, absolutely, no problem. So we pivoted to this. Could we, at least from our perspective as the evolutionary roboticists, everything that we as a community had done over the last 20 years, which you've now heard quite a bit about, could we redo the same thing with this stuff? Okay. All right, so where did we start? We started here. You've already seen this figure before. This was work, again, from Professor Cheney back in 2013. So my PhD student at the time, who took this course actually, he sat down and coded up HyperNeat, which, remember, paints patterns within a three-dimensional structure. In Nick's work, the different colors represented different material properties of soft robots. So at this point, we said, well, what are the colors in the HyperNeat going to represent if these cubes aren't soft silicone voxels, but they're cells, or at least small collections of cells? We didn't know, so we went back on the next Zoom call to talk to our Tufts colleagues again. And Doug explained at that time, in about 2018, he was very good at extricating skin cells from developing frog embryo. And he could also liberate myocardiac cells. So these are basically heart cells, which spontaneously increase and decrease in volume. Sound familiar? The myocardiac cells that make up your heart, exactly the same thing. So myocardiac cells in a developing frog, they form up, this is a human heart, not a frog heart. They form up into a frog heart, and they figure out a way to synchronize. There's a way in which all of those cells say, and get bigger together, and get smaller together, bigger together, smaller together, and so on, which is good news for the frog. Same thing happens in you. All of those cells synchronize. All of the cells that make up your heart get bigger together and smaller together, which ends up in the walls of your heart pushing outward, or all of the cells of your heart contracting inward, which is what makes your heart a pump, which is very, very good news for you. One of the most common things that go wrong with hearts is desynchronization, or atrial fibrillation. Actually, the world expert on atrial fibrillation is right across the quad here, Peter Spector, one of our collaborators. Anyways, we're going to work with hearts in this first Xenobot experiment. So the minute Doug told us that he could extract myocardiac cells and they get bigger and smaller together, we had it. So in Nick's experiment that you saw, there were 4 different cell types. There were green and red that got bigger and smaller in antiphase to one another. And then there were the light blue cubes that were passive and soft. We assigned light blue to represent frog skin, because frog skin, like human skin, is passive and soft. It can be pushed and pulled by muscle and heart tissue. So the only modification we made to HyperNeat was to reduce this output neuron from outputting 4 possible integers, the 4 different colors in Nick's work. We reduced it to 2 possible colors. Light blue, and you're going to see voxels in a moment that flash red, green, red, green. Green represents the myocardiac cells getting bigger. And when they turn red, that's them contracting. So far, so good? Having made that change, what is HyperNeat painting into our simulator? Oh, I forgot to mention, we're going to use exactly the same simulator that we used for the soft robot experiments. Soft cubes, which are going to represent bits of cells. What is HyperNeat painting? It's going to paint blue and red-green.
Like a cube robot, or not, maybe not even cubes.
maybe not a cube. So it's still going to, the HyperNeat, like in Nick's experiment, is going to paint a 3D geometry inside our empty cage. And then it's painting what across this 3D geometry?
Different colors.
Different colors which represent skin and heart. Yeah? So what you're going to see in a moment is different CPPNs. Remember, that's the name of the neural networks that HyperNeat evolves. Each HyperNeat is going to dictate a 3D geometry for the Xenobot. And then the colors are going to say, put skin here, put heart here, put some skin here, put some heart here, put some skin here. Yeah? It's dictating now not different material properties, but different cell types. Yeah? So geometry and cell type distribution. A very good way to describe your physiology is 3D geometry and cell type distribution. A lot of neurons up here, a lot of muscle in here, some fat also mixed in. Basically, it's dictating, HyperNeat now is going to dictate different kinds of physiology. Simple physiology, but physiology nonetheless. Okay, here we go. You've seen many of these. Before. Okay, here's our evolutionary algorithm. In this case, we started with a population of 100 random CPPNs, like we've seen before in HyperNeat. And you're looking at the results, the things that are painted by the first 3 random CPPNs in the population. Observations. What's happening?
I thought they were supposed to be synchronized.
Good point. The red-green cells are not synchronized. So Doug gave us a little crash course in frog heart physiology and said if they form up in the shape of a frog heart, they learn how to synchronize and do their thing. So as dumb roboticists who don't know any biology, we said, well, what happens if they don't form up in the shape of a heart? Because that's exactly what HyperNeat is going to do. HyperNeat is going to put heart tissue in weird places. Will those heart cells synchronize or not? And Doug's answer was, I have absolutely no idea. No one's tried this before. So what did we do, given that information? We deliberately made things hard on the evolutionary algorithm. We built in some more details here. We told the simulator that no matter where a heart cell is placed, just give it its own phase offset. So it's a little difficult to see, but you might notice that they're flashing red-green at about the same frequency. So I'm going to put lowercase t on the horizontal axis here. So now we're not talking about evolutionary time. We're just talking about time steps in the simulation. So any given heart cell in any one of these robots is assigned a sinusoidal pattern. It's kind of like a CPG or central pattern generator that we've seen before. It gets that particular cell gets bigger, smaller, bigger, smaller. The next heart cell that the HyperNeat paints gets assigned a random phase offset from the first one. The third one gets assigned another phase offset, and so on. So we're basically forcing the heart cells in the simulation to be desynchronized because we don't know what's going to happen. We're deliberately making things hard on the evolutionary algorithm. Okay, other observations? Nothing too surprising here. We've seen HyperNeat before. We create random CPPNs. These are the kind of things that it makes. What do you think the fitness function is? If you don't know the answer to this question, you have not been paying attention in this class.
Move to the right.
Move to the right, exactly. How well are these 3 robots doing at moving to the right? Not very well, right? Okay. This one down here, I think we might have mentioned this when we talked about Xenobots before. This one, it's hard to see. It's moving a tiny, tiny, tiny bit to the right. It's a little bit less crappy than the other 2. You know how this goes, right? These 2 get deleted, and the surviving CPPN produces a randomly modified copy of itself. And when that child CPPN paints a simulated Xenobot, it tends to have kind of the same 3D geometry as its parent and kind of the same cell type distribution, but not quite, a little bit different. Yeah? So very much, at least on the simulation side and the evolutionary algorithm side, very, very similar to things we've seen before in this class. I mentioned before that these soft-bodied simulators are pretty computationally expensive, so we had to run this on the supercomputer over in Colchester. And back in 2019, it took us about 2 weeks, and we got back this result. You're looking at 100 evolved robots. We actually ran HyperNeat 100 times. We were just talking about this with A/B testing. So let me go back to evolutionary time on the horizontal axis and fitness on the vertical axis, where higher means moving further to the right. We ran HyperNeat once, and we got this robot that moves to the right. It's a little difficult to see. There's a little rainbow trajectory underneath. This is showing how this particular robot moves to the right. It starts blue and ends red. We went back, created another random population of 100 CPPNs, ran it again, and got this robot, which also moves to the right. And we did this 98 more times to get these 100 Xenobot designs. They're all red. Why are they all red? There's no light blue. What happened?
They're all the heart cells.
It's one big ball of heart tissue. No skin whatsoever. This is maybe perverse instantiation, but kind of not. Again, we said make something that moves to the right. The move stuff is heart tissue. It just made one big ball of muscle or one big ball of heart. We're roboticists. What do we know about biology? We said, Doug, can you make any of these under the microscope? We knew that Doug could make these kinds of things out of skin. Could he make these kinds of things out of heart? And his answer is, I can't. And we don't have time to go into why, but the ability to do this microsurgery under the microscope is complicated. He said, I can't make all heart. I need some of the skin to be a scaffold. I need to be able to put heart on top of or next to skin. So we went back and made a change to HyperNeat, which is you're not allowed to paint all red. If you do, you get a fitness of 0. You have to include some skin. We did another 100 runs of HyperNeat, and it still fought us. In some cases, it says, I don't care. I need to be able to make stuff out of all muscle. But in some cases, it actually discovered mixtures of passive soft material, the skin shown in blue, with heart. We showed these 100 designs to Doug, and we said, can you build these? And he said, maybe, a few of them. We said, can you be more specific? So we sat down with Doug, and we won't go through this whole, table here, but basically we made this little thing, which is there contiguous tissue regions? Yes or no? What's a contiguous tissue region? There's a contiguous tissue region, there's a contiguous tissue region, this isn't. So HyperNeat can, in theory, put one skin cell next to one heart cell, next to one skin, next to one heart. Doug said, I can't build that. I need big blobs or patches of contiguous material. That I can build. So we started to create this little lookup table for Doug, and he went through each of the 100 and said no to some of these. So for example, where's our example here? This doesn't really have contiguous tissue, this one either. These ones got knocked out of consideration. The others, we went on, do they have a stable geometry? So little technical detail, gaps of about 100 microns. So where's a gap of about 100 microns? There's a gap of about 100 microns. Gap of 100 microns or larger represents the smallest internal concavity, which was stable at the time of shaping. So he couldn't make like a very fat donut with a very small hole in it. That was very hard to make. So he could do holes, but the holes had to be very large. So this one, it's a little difficult to see. It's got a little tiny puncture in it. He said, I can't build that. So that one got tossed out. This one got tossed out because it's got a tiny concavity. It's basically a filtering algorithm. So now we're down to about 50 or 55 possible things he could build. Then he's looking to see whether there's mostly passive tissue. We argued, dug down, he said, fine. As long as there's about 50% skin, I think I could possibly build it. This knocked out a whole bunch of designs because you can see that there's mostly red here. What did we end up with at the end? We ended up with 5 out of these 100 that Doug thought he could actually build. Yeah? So you know what's happening now. We're starting to cross the sim to real gap. We haven't even started to cross the gap yet, and 95 out of these 100 evolved designs have already fallen in the gap. We only have hope now for 5 remaining out of these 100 designs. We let Doug pick which of the 5 his gut told him that he thinks was most likely to be buildable, and he chose this one. It's got little stubby heart legs on the back, and the top half of this robot is all skin. Here it is. It does indeed move to the right. It moves to the right using desynchronized heart tissue. Not an easy design task. If you ask a human engineer to build a machine that moves straight to the right, and they have to build it out of motors that don't synchronize, this is a very difficult thing to do. But for Hypernate, it seemed to figure it out, at least in simulation.
So they're not synchronized. Are they not synchronized in a specific fashion? Like, you know, because if there was sort of, if they're completely random, then it makes sense to me that you could replicate it by just putting completely random ones in there. But if there's like, if you were to take that same ones and say, shift a quarter of them back by whatever and a quarter of them up by whatever,
yeah.
Then maybe now you have some like the sort of phase offsets were working in a way that they wouldn't work together if you randomly built the robot. Like, did you run into any issues with that?
It's a great question. So the way to talk about this is to talk about noise and frozen noise. And actually, this might be important for some of your final projects. So frozen noise means every time we run this robot, every heart cell gets its own phase offset, and it's always the same. Every time we play back the simulation, we set the phase offsets randomly first, but then we bake them into stone. Every time we play it back, it's always exactly the same phase offsets. That's frozen noise. I would suggest for your final project, if you have an environment in which there's things that are set or placed at random, freeze it. Make sure that every robot in your evolving population always sees the exact same environment. Here, we did not use frozen noise. Why not? It's much harder on evolution because evolution, every time this or something very similar to this arises in the evolving population, it's going to get different phase offsets.
Well, I mean, like, if you stuck it at the same, like, when you cross over to the real, you can't just, I assume you cannot just set the phase offset there, unless you have no idea.
Absolutely no idea.
There'd be no point in training, oh, wait, this frozen set of noise is really good, but that's most likely not the noise that you're going to get in the real world.
Guaranteed to not be the distribution of phase offsets that we're going to get in the real Xenobot, for sure. For all Doug knew when we were doing this, he said, maybe they'll all synchronize. Again, nobody's done this, so who knows, right? We can't know what the phase offsets are going to be. So when we were evolving these, they had to evolve to be robust to different phase offsets. You can play this robot back with different phase offsets, and generally speaking, it will move to the right. Actually, you're going to see a plot of that exact thing in a moment.
I thought the Xenobots were like in water. These are like...
Okay. In water. So it's a little difficult to see here. This robot is resting on the ground. The other difference we made in VoxCAD, which is the soft body simulator we're using, is we reduced gravity. So again, I didn't know, but it turns out that frog skin and frog heart are neutrally buoyant. They're heavier than water. So a frog that doesn't hold its breath and just hangs out in a pond will sink to the bottom, like our Xenobot. So what you're watching is something that's kind of running along the bottom of the swimming pool. It's moving through water. I take that back. It's not moving through water, it's moving through a vacuum. We did not simulate water, but we did reduce gravity, so it's kind of lightly touching the bottom. And as you can see, these stubby legs are pushing down into the back, which is a big part of the solution here. That tends to move the robot forward. Yeah, so we're getting ready to cross the gap, and you're going to see us cross the gap in a moment. We wouldn't be talking about this experiment if we didn't successfully cross the gap. But before we do, any other observations about this robot? You can sort of see how these backwardly pointing stubby legs are helping with rightward travel, or forward travel from the point of view of the Xenobot. What's the skin doing on the top? Turns out it's actually quite important. It's not just a passive sort of chassis for the motors in the bottom half of the Xenobot. They're all desynchronized, the heart, but it's moving in more or less a straight line. How is that even possible?
It seems to be sort of maybe storing some of the energy in the front left corner and then redistributing it.
Absolutely. So you can, if you look carefully, you can see the passive soft skin on the top kind of absorbing the pushing and pulling from the myocardiac tissue, the heart tissue underneath. And it's kind of storing or averaging or smoothing out all the little impulse forces that are pushing and pulling from below. It's kind of a smoothing averaging, which is taking all of that random movement and channeling it somehow into more or less straight rightward travel. Remember back to almost the beginning of the semester, we spent quite a bit of time talking about embodied cognition, right? The body matters. So as someone who's interested in embodied cognition, I was excited just at this solution alone. It's kind of showing how some of the idiosyncrasies of biology, in this case, desynchronized heart tissue pieces, can be helped by having the right kind of body. The skin is actually doing useful work here. There's no brain, there's no neurons. We are finally throwing off the shackles of neurochauvinism, right? This is just body, no brain.
The skin kind of reminds me of when you're showing the robots within robots research, the connectors that's raining in the different robots.
Yeah, it could be a version of that too. Yeah, exactly. The skin is kind of loosely coupling different parts of the heart tissue. Good observation. I hadn't thought about that before. Okay, anything else before we go on? All right, here we go. Let's try and cross the gap. Okay. As you can imagine, the gap here is pretty considerable. So let's switch over now and look down through the microscope with Doug. So here's Doug. He's actually building, this was when he was building this creature, not the one you just saw, but close enough. So he's taking some frog eggs, those are the big circles that you see, and he's injecting some dye so that he can determine cell type. So as these eggs start to grow into frog embryo, he's got to be able to find the skin cells and the heart cells because he's going to have to pull them out and use them. Here he is pulling off the vitelline membrane. So this is kind of some of the goo that surrounds the eggs. So we're watching Doug do a little bit of microsurgery here with very, very small surgical tools. So for scale here, the eggs are about 1 centimeter. If you go look at some of the ponds in Vermont right now, you'll see big clusters on the shore of those ponds of a whole bunch of these things, frog eggs. Okay. At this point, he's now removing the animal caps. This is one part of the egg at the north part of the egg, and that animal cap tends to contain, as the name implies, most of what's going to become the animal. The rest of the egg contains food, right? So he's trying to get at the animal, specifically the skin and the heart part of the animal. So in the next video, you're going to see not the eggs anymore, but these animal caps. Okay, here's a bunch of these caps, and he's separating the ectoderm. So this is the outer, the dark stuff is the outer part of the skin, which is not what he wants. And he wants the dissociated or the individual inner cells, the endoderm. This is the specific part of skin. So every one of these very small white dots that you see here now is an individual frog skin cell. You're not going to see the separation of the myocardiac cells, but it's more or less the same thing. So this is Doug basically starting to isolate and harvest the raw materials that he's going to build the physical Xenobot from. At this point, he's sucked up those individual frog skin cells into a syringe, and he's going to now deposit those skin cells onto this dark circle that you see here. This is a small concavity or well in the bottom of the Petri dish. We're still operating in room temperature fresh water, which is where frog is most happy. And these skin cells are settling into this depression and forming a thin layer of skin. Okay, he's going to do this multiple times. As he's doing this, what he's going to build up is he's going to build up this shape layer by layer. This is kind of like making a sandwich. When he was building this, he puts down just one layer of skin, then another layer of skin on top, another, and another. When he's building the robot that you saw that was moving, remember the moving Xenobot has heart on the bottom and skin on the top. So in that surgical procedure, he isolates heart cells, puts them in the depression, so a little layer of heart, another layer of heart, another layer of heart, another layer of skin on top, skin, skin, skin. So he's going to deposit layers of cells to try and get this distribution of cell types about right. Nothing to do with the 3D geometry yet. Okay. Turns out when you put the skin cells and the heart cells into this depression, they tend to settle into the depression, and then they also seem to be moving around like they want to connect to their neighbors. Think about the cells inside a frog and the cells inside of you. They're generally surrounded by a whole bunch of other cells. If you're a cell, the number one signal that you're in trouble is you have no neighbors. So our biology colleagues are pursuing this question of the fact that the raw material we're trying to build the robot from is kind of cooperating in the assembly process. Imagine you're building a car out of metal and plastics and ceramics, and those parts don't like to be on their own. They're actually coming together and trying to glom together. Maybe not necessarily in the way we want them to, but at least they're self-assembling. So another big important difference when we build autonomous machines out of biological materials rather than technological materials. The materials themselves have a mind of their own. They have ideas about how they'd like to be put together. And presumably, they also have ideas about how they'd not like to be put together. Okay. After many layers and pulling this thing now out of this concavity in the bottom of the Petri dish, you can see cells sphericalizing here. Again, they like to have as many neighbors as possible, so they're all kind of pulling inward. I think I mentioned this before. These cells, which are taken from very early frog embryo, they're quite adhesive, they're quite sticky, so they're sticking to one another, and then it seems they're also actively pulling and pushing to compactify or sphericalize this mass. So you get a big ball, a 1 millimeter sized ball of skin and heart. And now Doug goes back in. And in this case, when he was sculpting our Xeno sculpture, he's going to go in and now start to build the 3D geometry. And he's going to do this using one of the oldest art forms that humans know of, which is sculptor. Just scrape away the unwanted material. So he's using some very small tools, one which is like a very tiny cigarette lighter. It's a very, very hot wire, and he's cauterizing or burning away, burning a cross pattern into the sphere to start to approximate the 4 stubby legs of his Xeno sculpture. Here he is widening those cauterized channels in the material. So a mixture of sort of cutting edge technology and Stone Age art form here. Okay, here he is after about another hour of sculpting. I think I might have mentioned this before. Doug has very, very steady hands, no caffeine while he's working. Imagine trying to carve a pattern into a poppy seed. That's what you're seeing. Okay. At the end of this, Doug takes this Xenobot, turns it upside down, lets it float in the medium, and then he took this picture. Okay. That's the surgical process. As I mentioned, basically the same thing with skin and heart to produce this thing. I'm going to start this video in a moment. This is Doug's best attempt to get this cell distribution right and this 3D geometry right. You can already see from this image that the 3D geometry isn't perfect. We're about to try and cross the gap. There's already some inaccuracies here. But despite that, again, we wouldn't be talking about this lecture if it didn't work. Let me just put it on loop. Okay, let me play that again. How are we doing? You can see the little oscillation, right? That little oscillation is coming from the myocardiac cells doing what they do, which is get bigger and smaller. We wondered whether this robot was moving to the right because maybe the Petri dish is sitting on a table and that table has a decline of 5 microns. Remember, we're working with very, very small values, small things. So we wanted to be sure that this thing was moving to the right because of its shape and its cell distribution. So instead of putting it on a whole bunch of different tabletops, we just took this Xenobot and flipped it upside down. So it's on its back. You're going to see it on its back in a moment. Here it is right side up. Looks like a piece of popcorn. We took it and flipped it upside down. In a moment. Doesn't move. So it's not the table. It's not moving to the right because of the table or because of something else in the environment. It's because of the 3D geometry of the Xenobot. Question?
Where does the cells get like the energy for this?
Where do the cells, the myocardiac cells, where do they get the energy to contract and expand?
I assume there's like food in the petri dish that the cells.
There's a little bit of food in the petri dish, just like random bits of stuff, biological matter floating around in the water. It's not enough to do this. This thing is 50% muscle. It's an energy hog. Where's the energy coming from? I know we're not all biologists. Big hint. Remember where this thing just came from. These cells were just extracted from the egg. Why do we all like to eat eggs? At least most of us. Lots of energy. Where in the egg is all the energy? For some of us, too much energy. Not the white. The yolk, right? The yolk. It's golden colored for a reason. That's the gold in terms of energy content. This poor animal here, these cells, one of the things they were doing just before they were extracted from the egg is ingesting yolk. They pull little bits of the yolk called egg yolk platelets into the cell. So the tiny, tiny bits of the egg at the subcellular level. This animal was gorging itself on yolk just before the cells were extracted from the egg. And it's digesting that egg, that yolk, to produce the contractions.
So I guess at what point, do you know at what point it starts to run out of the energy from the yolk?
We know exactly at what point it starts to run out of energy with the yolk. It's the time at which the thing stops moving.
How long is that generally?
7 to 10 days. Okay, so it's a little bit of like a wind-up toy, right? There's a whole bunch of energy stored up inside. The skin and the heart cells do their thing, and then that's it.
Since it was like making itself into a sphere earlier, does it not want to like reshape after becoming?
Great question. So it's possible that the cells here are not happy. They've been sort of sculpted and burned away into this 3D shape, which is very, very far from frog, right? So this is not the situation they normally like to be in, but for whatever reason, they're able to hold it for about 7 to 10 days. It's possible that they're fighting it. So after 7 to 10 days, these cells themselves have gotten older. They've lost some of their adhesion. And like old people everywhere, they get kind of set in their ways. This isn't ideal, but I've got my yolk platelets. I got some neighbors. Okay, possibly. Still a very active area of investigation. I mentioned that the cells have their own ideas about what they do and don't want to be doing. If we're fighting that too much, it's probably not a good plan, but we're still trying to figure that out.
Have you tried putting more yolk in the dish to see if it would extend its lifespan?
So can we extend the lifespan of the Xenobots by feeding them more yolk? The answer is it's complicated. We haven't published on this yet, but yes, our Tufts colleagues have found a way to greatly extend the lifetime of these machines. It's not published yet, so I can't really give you the details, but the answer is yes. My PhD student, Sam, who did all the VoxCAD stuff and spent a lot of time with Doug, sending MP4s to Doug saying, build this, build this, build this, and Doug saying, I can't. Wait a second, I think I can. November 2019, Thanksgiving break, I was in my office on the far side of this wall here, and Sam came in and on his phone showed me this exact video. Science is hard. 99.99% of the time, the things we want to have happen don't happen. You're going to experience that over the next 8 days. This was one of those times where this crazy idea, maybe we can build frog bots that actually do cross the gap. They crossed the gap. I remember all the strength went out of my legs, and I just got up and I went for a walk down this hallway. It was Thanksgiving break. There was nobody here. And just the implications started to settle into place that we could do this. Now, how far this can go, who knows? But turns out we can automatically design autonomous machines built from biological components. That was 7 years ago. We're still working on it. There's lots of new results. We have a spin-off company around this technology, so it's growing. We'll see where it goes. We got 7 minutes left, so let's come back to this initial experiment. We tried. I got very excited personally about this, but as a scientist, you got to be sure that what you're seeing is what you think it is. So what did we do? First thing we did is to take this virtual Xenobot. We've changed the color here, but remember it's still skin on the top, cardiac on the bottom. And we reran this virtual Xenobot back in VoxCAD 30 times. And as Fitz was mentioning, we altered the phase offsets. We wanted to see how robust the virtual Xenobot was to different phase offsets. And all of the pink curves that you see here, this is looking down in VoxCAD at the virtual Xenobot. And most of these pink curves move to the right. Some of them actually come back around to the left, which is not what we wanted. But at least in simulation, the simulated Xenobot seemed relatively robust to phase offsets. So that was good. Then we took the in vivo Xenobot, the thing that Doug built, and Doug put it right side up in the Petri dish 30 times and recorded how it moved. Those are the 30 blue trajectories that you see, and most of them moved to the right. You'll notice that the spatial scale on the axes here is in millimeters, right? So we've got about a millimeter-sized creature that's moving about 4 millimeters. And what I don't remember from this figure is how long it takes it to move that distance. If I remember correctly, it's about 1 body length per second. So 3 or 4 seconds to move like this. Okay, question?
Did you ever find out if the heart cells will synchronize out of the shape of a heart, or is it just random?
It still looks random. I don't have any evidence of synchronization yet. Good question. Okay, these results, however, still don't rule out the fact that maybe the physical Xenobot is sitting on a table that's tilted 5 microns to the right. So we did exactly the same thing again, but we took the virtual Xenobot, flipped it upside down, put it back into VoxCAD 30 times with different phase offsets, and most of the time it didn't go anywhere. Sometimes just by chance it moved a little bit away from where we initially placed it. As I mentioned, Doug took the physical Xenobot in E here, put it upside down 30 times, back in the same dish, on the same tabletop with the same microscope and so on, and got the 30 blue curves that you see here. So this was more quantitative evidence supporting the hypothesis that it's this shape and this cellular distribution that affords or gives this machine rightward travel.
So the image of the in vivo looks like it's more detailed almost, and I guess how many voxels are in the silico versus how many actual cells are in vivo?
Yeah, very hand wavy at this point. So Sam would send this video to Doug, and that's it. Doug was free to try and make something that looked more or less like this with heart on the bottom and skin on the top. As you can see, this is definitely not an exact science. It's still not possible to put individual cells exactly where VoxCAD says. So at the moment, and still to this day, a cube in VoxCAD means put about 1/10 of a millimeter cubed of cell type X here. A cube represents a bunch of cells, not an individual cell. We're getting closer to being able to do cell-by-cell assembly, but we're not there yet.
Have you tried scaling up the size of these at all, or is it staying relatively small?
Great question. So when we published all this, we got quite a bit of media attention. We started, among other things, to get emails from the general public. We had a 7-year-old email us and said, could you make a Xeno elephant? Could you make one as big as an elephant? We all had a good laugh, as you are now. And then Doug said, well, actually, there's nothing about this that says it has to be done at the micron or the millimeter scale. But anything above 1 centimeter suddenly gets much harder. Anybody remember your high school biology? What is it about the 1 centimeter barrier that's difficult? We've got skin and heart. Remember that they're busy munching and eating on yolk.
Because it lacks bone to support the body.
You need bone when you get far beyond a centimeter. Before that,
It's the surface area to volume ratio.
becomes a problem. Why? What happens? So the surface area to volume ratio starts to change as you scale up from the millimeter to the centimeter. But why is that problematic? You said because the heat can't get out. It's not the heat. We're not dealing with computers. What can't get out?
Just like a lot of energy consumption.
Getting closer. What can't get out?
It's like
carbon dioxide, right? The cells, if you make a 1 centimeter ball of cells, the cells in the middle start to suffocate, right? If we put 1,000 humans tightly packed around you, you'd also start to suffocate for exactly the same reason. So our answer to the 7-year-old was, if we can solve the CO2 problem and getting oxygen in and CO2 out, we could at least get beyond 1 centimeter. I don't know about an elephant scale, but not impossible. Okay, one more question.
If you were able to take the heart cells and form them into a shape of a heart, do you think it would then synchronize? And do you think then if you had made just a heart from that, you could use that to have synchronized action in something?
So if we figured out, or Doug figured out how to put frog heart cells into the shape of a frog heart, would it synchronize? You're asking the wrong person. We're beyond my area of expertise. I have no idea. Remember, that would also be very big. So constructing something of that size by hand, not possible yet. Okay, we'll continue talking about Xenobots in our final lecture on Thursday. You have a quiz due tonight, and you're starting to work on your written report and oral presentation. Thank you.